Dear colleagues, good afternoon, and I'm so glad to have this chance for second time in my career to present in HRI, our second actually study. So the topic of our study is the Ferrum phosphoricum D12 effect on mouse macrophage cell proliferation and transcription of inflammation, oxidative stress, and iron metabolism related genes in conditions of LPS, bacterial lipopolysaccharides, challenged macrophages, actually. So we conducted an experiment which was testing the ferrum phosphoricum in D12 potency, the Schussler salt number 3. As we know from the practice, is used in acute phase of inflammation for treatment of cold and fever, also used as a remedy in case of wounds treatment, burns, muscle fatigue, and last but not least, treatment of iron deficiency anemia, with the potential to increase the intracellular iron levels and inducing, respectively, the iron metabolism. So in this relation again, we need to keep in mind that macrophages, which are our target in recent experiment, you will see, we have majorly two types of them. You have M2 class, which are so-called classically activated ones, and we may name them the healers. They have a major role in tissue recovery, repair, after wounds, injuries, and they also found that they have a role also in carcinogenesis. But M1 alternatively activated ones, they are the soldiers. They are those that fight against the pathogens as a part of our immune response. They have a role in inflammation, respectively. Also, they are known with their activity to suppress tumor development. So these two types of macrophages expectedly need to have a different response to iron and different iron metabolism. So they should have change in expression of the genes for ferritin, ferroportin, or iron-dependent transcription factors, which we will see we also analyzed. Iron and macrophages has different relations. So we know from the literature that when the heme and iron are presented in the cultural media, they may shift the polarization of tumor-associated macrophages toward pro-inflammatory, tumor-suppressing ones. So from different point of view, so-called nurse macrophages, as macrophages as the cells which are involved in the recycling of erythrocytes, they may serve as an alternative source of heme iron for the new erythroblasts, respectively. They may develop iron or heme iron in different forms, papers show. I want to share you, in the end of introduction, the paper we published in 222 in Homeopathy Journal. Actually, this is the previous study. It was based on the same idea. We test the effect of ferrum phosphoricum D12 on cell proliferation and transcription of the same set of genes, but in non-stimulated macrophages. So once we see that there is an effect on gene expression, even in normal macrophages, without putting them in conditions of inflammation, we get curious, and we started the second study. It was now aiming to analyze the cytoprotective and immunomodulation potential of ferrum phosphoricum D12, ethanol preparation in vitro, but this time in LPS challenged macrophages. So we'll see what effect the ferrum phosphoricum has in conditions of inflammation. What does it changes in macrophage metabolism. So the first task was to analyze the potential cytoprotective effect of ferrum phosphoricum D12 in model of ethanol-induced cell death in mouse macrophages. Ethanol, we know from our previous own studies, also from literature, it's quite well explained that ethanol causes an oxidative stress and inflammation, respectively, normally expected leads to cell death, and we used that. Our preparation was done in ethanol respectively. And we used ferrum phosphoricum ethanol preparation as the main object, and we have a control of the same ethanol solution without ferrum phos, of course. The test that we use is the known MTT test that my colleague already told about it. I won't get in details, but the idea of MTT test of cytotoxicity here was to test the cytoprotective effect, of course, on macrophages, and to determine the working concentrations that we use in our second task you will see. So briefly about the experimental model. We seed the macrophages in a given concentration, and after 24 hours we treat them with ferrum phosphoricum ethanol preparation or ethanol as a control treatment. And then at the day 2 we measure the, by MTT test as you see, the cell viability. And here are the results. So I'll pay your attention to the first to the effect of ethanol, the orange line. You see with the increase of concentration of ethanol expectedly and dose dependently, we have a significant cell death from certain concentration. But here is the effect of, which is the more interesting, of ferrum phosphoricum D12. It completely neutralized the ethanol-induced cell death, and that's not enough. It stimulates the significant proliferation of macrophages up to 158%. So it completely neutralizes the toxicity of ethanol. And then here are the 3 concentrations we picked up for the second part of the second experiment, which second experiment was the model of LPS challenged macrophages. The same steps we have with the slight differences, of course. We see the cells in the same concentration. We put in the culture media ferrum phosphoricum, of course, or ethanol as a control treatment at the end of the 24th hour. Then the day 2, we treat them or not with bacterial lipopolysaccharides in order to stimulate inflammatory response in these macrophages. Respectively, at the 72 hours after, we might isolate RNA and protein for qPCR analysis or Western blot analysis of proteins. So here are the results in full form. I will first pay attention because there are many treatment groups. I know it's confusing the first shot, but let's start with the first group. You have a control group of untreated cells, normally expected. You have a group of lipopolysaccharides, which you see 5,600 times induces the gene expression of interleukin-1 beta, the cytokine of the acute inflammation. First of all, what we could say, the model works perfectly. Obviously, we have induction of inflammation. Second set of treatment groups. Here are the groups of treatment with ferrum phosphoricum and control ethanol treatments of macrophages which are non-stimulated with bacterial lipopolysaccharides. In next slides, I won't show you these results because they're repeated, which is nice. I need to underline this. The results that we get in our previous study. So we confirm our results in this study, the results that we get from the first one. And then, what was the new one and curious we are to see is the second set of treatment groups. You have 3 same concentrations of ferrum phosphoricum in macrophages which are now challenged with bacterial lipopolysaccharides. And of course, the treatment with ethanol control solutions, also in macrophages treated with bacterial lipopolysaccharides. If we recalculate the gene expression compared to control LPS treatment only, here is the focus. So when you treat with the ferrum phosphoricum macrophages, they induce even more, they intensify the effect of bacterial lipopolysaccharide on macrophages. Then compared to treatment with ethanol control groups, you have a statistical significantly higher effect for the gene expression of interleukin-1 beta. Actually, that's the best marker, and we might say that's enough. As a gene you tested when you check for the acute inflammation, but we continue with interleukin 6. In case of interleukin 6, we have known that nice difference with the ethanol group, but still we have induction of the interleukin 6 gene expression by Ferrum phosphoricum higher than the LPS treated control group again. Then about tumor necrosis factor alpha. Here it's much more clear, as it was with interleukin 1 beta. We know that the tumor necrosis factor and interleukin 1 beta are the major ones ruling the inflammation in the first stage, acute phase. And you perfectly see here that again we have a stimulation even more than just lipopolysaccharide do when you put the Ferrum phosphoricum in culture media. And again you have a significantly different effect that distinguishes the effect of Ferrum phosphoricum from the ethanol treatment. Another thing maybe you should pay attention. Do you see here a dose-dependent effect? No, you won't see it in other treatments with Ferrum phosphoricum as well, but treatments with ethanol give a quite clear dose-dependent effect. This, I think, is important from homeopathic point of view. Maybe you will say better. I'm not a homeopath, you know. For inducible nitric oxide synthase, an important enzyme from the inflammation. Again, you see the induction of the gene expression of nitric oxide synthase, the inducible one, which is related to inflammation, much higher than just LPS when you have a ferrum phos. And here you have one concentration which is significantly higher than its ethanol control treatment. The other ones are lower than ethanol. Maybe we need to repeat these ones especially. But what interesting we have, the second set of genes we analyzed, the antioxidant enzymes. We tested the gene expression of glutathione peroxidase, also of glutamate cysteine ligase. We have no statistically significant changes for glutathione peroxidase in case of LPS challenged macrophages. But when we talk about glutamate cysteine ligase, an enzyme, which is the main responsible one for the synthesis of glutathione, which is the intracellular antioxidant of the body, and also is a cofactor used by glutathione peroxidase to fight free radicals, especially hydrogen peroxide and lipid peroxide. You see clearly that you have induction of this enzyme by Ferrum phosphoricum, even higher than lipopolysaccharides again. And one more fact, this effect of Ferrum phosphoricum is different than the control ethanol treatments, which is quite nice. When we talk about iron-dependent transcription factor, here we see that Ferrum phosphoricum treatment reduced this time the transcription of this iron-dependent, the transcription of iron-dependent transcription factor. It was lower than the expression of the gene in the group treated only with lipopolysaccharides. Yes, the effect again was different significantly from the ethanol treatment. About ferritin gene, especially the ferritin light subunit, we have induction of the ferritin gene expression higher than LPS treated group, and it was also significantly different from ethanol. This is the marker that I liked most. I'll tell you why. This is CD68. It is a marker for differentiation of macrophages toward M1 phenotype, pro-inflammatory, tumor-suppressing ones. So it means that this marker should be higher when they get into a phenotype of pro-inflammatory macrophages. So we see that not only lipopolysaccharides were inducing this marker, but even more it is induced by Ferrum phosphoricum, significantly, and significantly higher than ethanol control groups, which was perfect. Let's resume all these results. First of all, we confirmed the previously reported immunostimulating potential of Ferrum phosphoricum, also the potential to has impact on iron metabolism in non-stimulated macrophages. The new results show that Ferrum phosphoricum possessed a protective effect against the ethanol-induced macrophage cell death. Ferrum phosphoricum reduces LPS-induced transcription of IRIP2, which is the iron-dependent transcription factor. But ferrum phosphoricum intensifies the LPS-induced antioxidant enzymes, inflammatory cytokines, and ferritin gene transcription. So what we might conclude from all of these, ferrum phosphoricum D12 may exhibit an immunomodulating potential, antioxidant and iron uptake potential as well in LPS-challenged macrophages by inducing the changes in related genes transcription. Of course, the results were strongest when we talk about immunomodulation and antioxidant effect. On my opinion, we need to do more tests to clarify the specific effect on iron metabolism. Another observation that we did was that the changes in gene expression in the transcription levels are being dose independent. So, if we speculate, we may say that thus, ferrum phosphoricum, preparing the macrophages for a stronger immune response, maybe it may shorten the recovery periods after infections. That's my hypothesis. From the practice, you will see how it looks like. I hope they will cover. What are the future perspectives? This was the analysis of gene expression on transcription level. But the next step is to analyze the protein expression levels. They are ongoing at the moment. We just had no time to finish these experiments and present them here, but we'll do it next time, I hope. In the end, I want to thank my colleagues. We did this work all together. Diana Vankova, Milena Pasheva, also chief assistant professors from the Faculty of Pharmacy in Varna Medical University, and Professor Kiselova, and our supervisor, Professor Ivanova. And our supporters from DHU, which prepared especially these preparations of ferrum phosphoricum for us, and Alpen Pharma for their all support during all these projects. And thank you for your attention and hospitality. And in the end, welcome to Bulgaria again. It's also a colorful country.
Thank you very much, Professor Oskan.